If you step into a New Zealand forest, what do you see? This is a simplified model of an ecosystem, but we can kind of imagine a structure like this. There is still something missing though. Can you spot it? It's the parasitoids! Of course it's the parasitoids, that's why we're all here. Where do the parasitoids go? Well, they parasitize mostly other insects, but also some plants. A parasitoid is basically just a parasitic organism that doesn't actually inhabit the host itself, or only inhabits the host as a juvenile. If you think of a classic parasite, like a tapeworm, we all love tapeworms there. They're really cute little fellows. A tapeworm lives inside of you as an adult. These wasps don't do that. These wasps are free-living as adults, and they're not even necessarily feeding on the host animal. The reason they're parasitoids is because their larvae live in or on other living creatures. In any given environment, there'll be a bunch of insects and a bunch of plants which have a lot of juicy, delicious, fleshy tissues that you could lay your babies on, and the babies could enjoy. And so there's lots of parasitoids. They're often very, very tiny, and they're very, very common. Anywhere you go, you'll find them except, I guess, Antarctica. What if we had some mammals? That causes a huge amount of chaos. Our native animals are not equipped. They don't even understand that a ferret with sharp teeth is a problem. They're like, oh, hi ferret, and then they get munched to death. Okay, now we clear half the land, right? We humans have come in. Sorry, forest, you're half the size you used to be, right? Now that's dairy pasture. We're not putting any native owls on that field, right? That's where the cows go. Okay, now that's not good, right? We've lost half of our space, and some of our plants need a lot of space. So the ecosystem is looking not only really, really complicated, but kind of really screwed. But maybe it's not screwed. How can we even tell? Well, don't worry, because what if we add a whole bunch of junk? What if we add oil and fumes and plastic bottles everywhere? And we've got more invasive animals. And now all of the relationships that we thought we knew are totally rearranging because we've just added a whole bunch of new factors, and it's too complicated. How could we have predicted all of this? Every change you make has a knock-on effect to every connection that animal has to the entire rest of the environment. Oh my god. What do we do? This looks like an absolute nightmare. Oh, I should do like a thumbnail face, right? Like a... Anyway, sorry. I am interested in the parasitoids, but I'm also an ecologist. And what I'm interested in is just the ways in which we gain fundamental knowledge of what this picture looks like. So before we even start coming up with solutions or interventions, how do we know what's going on when what's going on is so complex and so dynamic and so constantly changing? Maybe we can use parasitic wasps as bioindicators in Aotearoa, New Zealand. Maybe we can't. That's the other possibility. Join me as I regale you with parasitic wasps. Biodiversity is one of these words that biologists can't agree on what it means. There was an interesting paper which was like, can we stop using the word biodiversity? Because it's kind of this vague catch-all term that just means how much different stuff is there. You can imagine like a desert is very non-biodiverse because there are like 3 animals there and they're all little ratty things. And there are 2 plants and they're both cactuses. And like the rainforest is like the most biodiverse place on Earth because there are like 100,000 species of beetles there and there are 50,000 birds. There's 50,000 tree species and there's lots of animals and it's crazy and it's productive and it's biodiverse. When I say it here, what I mean is basically just like, are there lots of different animals or are there not many? I kind of fall on the side of it's a dumb word, but it is the standard word for this kind of study, so we'll use it here. But if it gets confusing, or if you think, well, that's a bit vague, it is a bit vague, and I'll try to address it where I can. It bothers me too. The second idea is the idea of ecosystem degradation. So this is an ecosystem getting worse, but it's important to separate, I think, the morality of degradation from the empirical reality. It's really hard not to be like, oh no, it's a bad thing that this ecosystem is getting worse. There's some kind of duty to make sure that the ecosystem stays good and doesn't get bad. But this is human stuff, right? These are human values. But as a scientist, it's just change. And we just have to know what the change is and why it's happening. And we can we can leave deciding whether or not it's bad to people downstream. So when I say degraded, I just mean changed from the way that it currently is. Native things are being replaced by invasive things. Forest ecosystems are being replaced by urban ecosystems or pasture ecosystems, which are generally less diverse. Why do we need to monitor? Well, there's 3 categories of reason. The first is the pure science, because it's really, really interesting, and because the forests of New Zealand are so unique and have such a rich biological composition that there's just so much we can learn by studying them, and there's so much we don't know. The second reason is, if we're going to help something, if we're going to solve a problem, we have to know that the problem exists. In a sense, ecosystem monitoring is a job that is going to create more jobs. We can identify things, and then maybe we can solve them. The other thing is that the best time to plant a tree was like 4.5 billion years ago, when the Earth... Well, no, I guess that wasn't so good. The best time to plant a tree was like 600 million years ago, like end of the... What was it called? The cryostasis? The cryostasis era? Like coming into the Cambrian. Because there were no trees back then. There were like proto-plants that were like, didn't have solid vasculature or anything, but they were plantish. So if you planted a tree back then, you would have basically kicked plants forward like 500 million years of evolutionary history. That's the best time to plant a tree. But the second best time is today, right now. Not tomorrow, today. Similarly, if we go out and monitor an ecosystem today, even if it seems like that knowledge isn't doing anything, having a historical data set to compare to once climate change really starts setting in is going to be really, really important. So ecological monitoring, I think, is the backbone of any biological work we want to do in the field. How? So how do we do it? I mean, the simplest way is you go out into the field with a clipboard and a pen, and you say, that's a tree, that's a pigeon. You can see I've said vegetation counts. Vegetation counts. When I say vegetation counts, I mean it literally. I mean going into a forest, deciding on like an area, and counting the number of each kind of plant that you see. And that turns out to be really monotonous and takes ages, but it's as good a way as any at finding out what's out there. And you'd be surprised at how much you can learn from counting trees. They say that the only difference between science and like just looking at plants is the fact that you're writing it down. But there's more complicated things you can do. You can gather physical material. You can take plants and animals out of the environment, take them back to the lab, see what they have going on. You can take samples of abiotic stuff, like soil and the air and the water, and you can run DNA tests on your samples to see what the microbial community is like. eDNA is really taking off. eDNA is this idea that you don't actually need a particular animal. You don't have to find a mouse and take it back and sequence that mouse's DNA. You can just take a bucket of water from a river and sequence all the random fragments of DNA you find. And you're not going to construct a genome of an animal, but you will construct a kind of collage of random coding sequences, which, if you have a lot of computational power and the right theoretical tools, you can probably analyze. But eDNA is in its early phases. And finally, what can you do? You can put out traps. Like a villain in a Saturday morning cartoon, you can put out a pitfall trap. And in fact, that's what we do. Humanity has been around for 600,000 years or something. And, it's been 2,500 since Plato, founder of life. And the best we can do is a pitfall trap. So what we do is you get a pot and you fill it with antifreeze. You bury it in the ground so it's flush with the ground. And then a bug walks along your trap and it falls in and it dies in the antifreeze. And then you leave that out for a few days and you come back and get it and you get a pot, and in the pot is antifreeze slurry and assorted insect goop. It's great. Yeah, that's an important tool. And in New Zealand, the Department of Conservation is putting out thousands of these every year just to find out what kind of bugs are in our forests. Now, you may have noticed, it does kill the bugs that it catches. Okay, So I kind of want to do this. I want to put out a trap, but I want to target just parasitoid wasps. Why? Well, to understand, we have to talk about a bioindicator. A bioindicator is any living thing that you can use as a proxy for wider ecological biodiversity. So when I say proxy, I mean you can measure the bioindicator, whatever it might be, and the status of the bioindicator gives you reliable information about everything else that's happening in the background. We think, parasitoid wasps are a great bioindicator for terrestrial ecosystems that are going to help with the water because they live in forests, but forests don't have a bioindicator system at the moment, and rivers do. Rivers have something called the macroinvertebrate community index. The way we determine what the water quality is by using a bioindicator. We use freshwater insects, macroinvertebrates, so that just means big bugs, basically they live in the water. And if you have a bunch of big bugs that are pollution sensitive, so they die in pollution, but they're there in your lake, good, you're not polluted. If you don't have them, sorry, you get a bad score on the MCI. We want to make a terrestrial version, a macroinvertebrate, a macroparasitoid community index for trees or something. And we want to do it with these guys. So these are photos I took in the microscope at my lab. I particularly like this guy. He's pretty cool. These are photos under a microscope. So I've trapped the wasps, I've put them in a petri dish, and then I've put them under the microscope, and this is what I see. These guys are mostly diapriids. You can tell the wasp is a diapriid because it has this sort of shelf. If you look at the big guy there, you can see that he has the kind of head, and then he has this antenna coming out, and he has a kind of shelf-like bar, like he has a big flat nose. That's characteristic of the diapriids. Whereas this one down here, this one, doesn't have that. It's just got like a round head, and that's a chalcidoid wasp. They're certainly not like the big wasps that you are probably used to. So they don't have a stinger. The big spike thingy, where their stinger is their ovipositor, so they lay their eggs through it. But they can't sting you, they can't hurt you. They can inject poison into their victims with their ovipositor, but they're so tiny that it's not going to have any effect on the person. It won't even be able to puncture your skin. I could choose any animal in the forest to be a bioindicator. Why choose parasitic wasps? They have 3 strengths as a bioindicator. The first is they're diverse and they're abundant. So if you put out an insect trap in the forest, you will almost always find a parasitoid. I put traps out just around the clock tower at my university, and there's lots of parasitoids. I've put them out in my garden. I live in I won't dox myself, but I live in a urban area, right? Like it's motorway outside with cars zooming by, and there's forest fragments, but it's not that nice to be an animal. But there's heaps of parasitoid wasps in my garden. They're everywhere. So if we wanted a measuring tool that we could just deploy as needed to any ecosystem, parasitic wasps, we're very unlikely to run into a case where it doesn't work just because there are no wasps at all. The second reason is that they're host specific. So they can't just parasitize any old thing. They have a very specialized ovipositor, like the egg dispensing mechanism, that will target one or a few different types of host. The main 2 hosts they target are caterpillars and beetle larvae, so little grubby things, and they will come up to the host and inject their eggs into it, and the eggs will either hatch inside the host and eat it from the inside out, or hatch flush with the skin of the host. So you have the host, and then the egg kind of sticks out like this, and the larvae goes for a ride for a while on the caterpillar as it crawls along the plant, and then it starts eating the caterpillar. So that's really good for a bioindicator, because if we find a parasitic wasp in an environment, and we know which bugs it parasitizes, then we know that the hosts are also there. So we can catch a bunch of wasps and then say, well, this wasp parasitizes that caterpillar, and this one parasitizes that beetle. Now, we didn't get that caterpillar or that beetle because they're rarer and bigger and lower, and we're less likely to catch them. But we know they're here because we found the wasp. And that's a bioindicator, right? They are at least a bioindicator for their host. And finally, they occupy a very high trophic level. So that means that on the food chain, they're right at the top. So trophic levels are like, if I come back to this diagram, the lowest trophic level is the plants, right? They just get their energy straight from the sun. And then we can go upper trophic level to the herbivores, and then upper trophic level to the animals. And the reason you want your bioindicator at a high trophic level is that if you're all the way up the top, up here, you can only exist here if every layer below you is functioning. So you're relying on the prey to be there for you, and the prey is relying on the tree. So that's the idea. The wasp needs its host. It needs the things it's eating. It needs all the things that the host needs to survive. So that's the background. That's the kind of first half of the video. I need to go to bed because I'm really tired and I've taken a medication that makes me sleepy, and I'm falling asleep. You can probably tell by the way my excitement is fading. But I'll be back in the morning with the second half, where I'll talk about the details of particularly what I will be doing and what I hope to achieve. Thank you, and see you in a moment. So the project has 4 questions, and they're all working together towards the general goal of seeing if parasitoids can be bioindicators. The first question is, can we use data from parasitic wasps to diagnose wider biodiversity in forests? A second question that we can use to hone in on this, to get a specific correlation metric, is, does vegetation diversity correlate with wasp abundance or diversity? So in this case, abundance means just the sheer number, and diversity means the number of different species. So the question is, do we find more wasps or a more diverse range of wasps in places with more plants compared to places with less plants? It's kind of a crude measurement. It would only tell us so much without knowing what kind of plants those are. But if it's true, that gives us a good reason to think that the project is viable. Third question, what subset of insects do different trap types catch? This one is important because currently, when you use a pitfall trap, you just catch any insect that walks on the ground. They are really non-targeted. They'll just catch anything, it'll fall in the pit and die, which means you can end up killing a bunch of insects that are rare or native, like really culturally important. It's dangerous. Whereas for the wasps, we can use a trap called a yellow pan trap. And finally, how can machine learning reduce the cost of wasp monitoring? So one of the big pitfalls of moving away from pitfall traps and towards wasp traps is, well, these wasps are very, very tiny, and they all look pretty similar, and they're very, very diverse. They're not the kind of thing that you can expect someone in the field who isn't trained to identify. That you need a microscopic camera, and you need some kind of taxonomic expertise. We have keys. It's kind of like a choose-your-own-adventure book, but with a series of photos of wasps, and you kind of say, how many segments does its antenna have? Is the shape of its ovipositor X or Y? And then you follow the path and you end up with a diagnosis. But they're quite time-consuming, and they can be pretty complicated. What would be better would be if we could make some kind of photo ID app. Something that you take a photo of the wasp, and then you send it to the machine, and with relatively high confidence, it can tell you that's a chalcidoid, that's a diapriid. If we could get it even more refined, it would give us like a genera rather than just a family, that would be good. But even just a family diagnosis could still give us something. So those are the questions. Now, how is my research going to answer them? Well, what we'll be doing is laying out traps in a range of regional parks across Auckland. Editorial note, it's not actually going to be at the regional parks because I was sick for a whole year and we've changed. We're working with a different group now, but the methodology remains the same. In each of the kind of regions that we are sampling, we're going to look for degraded forests, regenerating forests, and old growth forests. The idea is that each of these sites we can diagnose quite easily by kind of looking at it, and we know from a bunch of other data that there's a correlation between these types of site and the vegetation diversity. So if we put traps out in an old growth forest, we can see and we can have high confidence that the vegetation diversity is very, very high in that area. In a degraded forest, it's very low. So we will go to our sites and then we'll measure plant diversity and a bunch of abiotic conditions. We want to know light levels, temperature, what the weather's been like, soil acidity, a whole range of different things. And we'll put those into a data set. Thankfully, wherever we end up working, a lot of this information is publicly available. If you're an entity invested in some kind of conservation, you'll usually have ecological data that you've gathered for other purposes that we can use. Here is the experiment. For each site, we will put two kinds of traps out. We have the yellow pan trap and the SLAM. So a yellow pan trap is, as I described before, it just looks like a yellow kind of box. Actually, I'll go get one. So, here we go. This is a yellow pan trap. Wow, the colour is quite different on the camera, but as you can see, it's very yellow. This one's been out in the dirt, but it's just like a yellow plastic tub. It's about as simple a piece of technology as you can imagine. I think they just 3D printed these. They're cheap, easy to get, and they work. And yeah, so all you do is you pour water in. What is this? You pour water in, pour a bit of detergent in, put it in your region, and then come back in a couple of days, pick it back up, and you'll have a nice kind of bath full of debris and plant matter and bugs, hopefully. The biggest problem with them is that if it rains, you're a bit screwed, because it will just wash everything out. So you do have to time it so that you put them out when it's dry. Yeah, and so you can put out heaps of them and then just take them all back in. You just pour the contents into a sieve and then kind of extract all the bugs and then put them into a tube of ethanol. So you just preserve them in 95% ethanol. And then, cool, now you've got a tube of bug from your trap. So that's the yellow pan trap. The other thing is a SLAM trap. Now SLAM stands for Sea, Land and Air Malaise. So a malaise trap is a trap named after a French guy called Malaise, and it works by having this kind of net that bugs fly into and then get trapped in. It's a sea, land and air trap because they are kind of multi-terrain. You can float them on the water, you can just put them on the ground, or you can hang them from a tree branch and they'll kind of catch flying things. So they're very versatile. They are, for better and for worse, much more effective than pan traps, in the sense that they'll catch way more stuff. Now sometimes you want to catch way more stuff, and sometimes you don't. There are definitely pros and cons to killing a lot of bugs for science. We run this study from December to April. Now, as you can see, it says 2023. Plan was for this to run last year, but then I got sick, so I couldn't do it. So we've had to put it off a year, but we're starting now, in about a week. We run it over the summer months, December through April, because so I'm in New Zealand, southern hemisphere. We're coming into summer. It's like late spring now. That's when all the wasps come out. Spring is when like life returns to the fields, and then the parasitoids have to wait for the baby insects to come out, and so they have a bit of a delay. They come out usually in like the start of summer to late summer. So December to April is our trapping regime, and we put the traps out in a grid like this. So we have 12 yellow pan traps scattered, separated by 5-meter intervals around a site with a SLAM trap dead center. And the reason that we have so many traps is because there can be a lot of local variation, like variation over the span of like 10 centimeters in what bugs fly in, and we're going to need a big data set to kind of see past that into the whole environment level. So we leave them out for 5 days. We repeat for 5 months, 5 replicates, and we put out 13 traps per site. We're going to end up with somewhere in the ballpark of 500 wasp samples. That's a lot of wasps. My next few months are going to be primarily moving wasps from vials into petri dishes. You've got to label each one so you can keep track of where they are. So you need like a field code. I'm working on that right now. I might put out a field code video next week, because the field code is one of the first things I'm going to need. I'm technically still shackled by the university because I had to put my degree on medical suspension. I'm not allowed to do any work on the degree until December 1st, which is in 3 days. On December 1st, I'm unleashed. I can do my writing, I can start my planning, I'll be all guns blazing. Once we've got that, we've got all of our samples, we take them to the lab, and we get something like this. So this photo here, is an actual petri dish of a yellow pan trap that I took in. So that's just a single pan trap. And you can see we have quite a few wasps. They're the kind of big black guys. I know they're pretty small on the screen. Quite a few wasps. We have some flies as well, some little flies. And then we have the big white things are amphipods. They're like land crustaceans. They're like little shrimp that live in the soil, basically. So this trap got quite a few amphipods, and then I think that's a snail at the top. We caught a snail. These are what my vials look like. Those are my old test labels. So the idea is we pour everything to the petri dish, and then I'm going to go through with forceps and separate the wasps from the non-wasps and leave the non-wasps in a vial of non-wasps for someone else to deal with. And then I'll take the wasps out, I'll photograph them, I'll ID them, and I'll record everything I can about them in a spreadsheet. What I want to do with my wasps is identify them to some kind of recognizable taxonomic unit. Now, okay, that's words, right? What's a recognizable taxonomic unit? If I were dealing with birds, say I had put a bird trap out, a yellow bird trap, and I had like 600 birds in a petri dish the size of this room, and I had like massive forceps, I was like, oh, I'll pluck the pigeon out and put it in the pigeon vial, right? In that case, I would be identifying my birds to species. I want to say, this is a pigeon. It's belongs to the species of like rock dove, which pigeons are, or this is a red-billed seagull, or this is a sparrow. Very easy with birds. You can identify them to species. With wasps, the taxonomy is so complicated because there's so many wasp species and they all look very similar, that it's not really practical to ID them all to species. Some of them, there'll just be no way to know. Like, we won't have a species name for them. They'll just belong to a genera, and maybe they're a species that we have, or maybe they're a new species that hasn't been named before. Someone would know, probably. I could call up some entomologist, but I don't have time to do that for every single wasp. So what we want to do is come up with some kind of higher level taxonomic unit. We say, this wasp belongs to this group of wasps, which are all more related to each other than they are to wasps that aren't in the group. So it's a clade, but it's not necessarily a species. There might be multiple species in the group. It's just that they cluster together evolutionarily, so I can designate them as a single unit. Once I've done that, so I've got all my wasp data, put it in a spreadsheet. The next thing to do is quantify the abundance and diversity. So, I mean, abundance is literally how many wasps did I find, like count the wasps. Diversity is how many different units have I got for each site. And then we're going to do a bunch of stats. So we'll have a big Excel spreadsheet, we'll have RStudio, and we'll just run some statistical tests, see if we can find a correlation between wasp data and plant biodiversity, insect biodiversity across sites. The null hypothesis, it's always important to state your null hypothesis. A null hypothesis is the prediction that there'll be no effect of whatever you're trying to test for. So in our case, the null hypothesis would be there is no correlation between wasp diversity and wider ecosystem biodiversity. It's just random. The wasp will go up and down, and the ecosystem will go up and down, and they don't correlate. So we can't do anything meaningful in terms of ecosystem diagnostics. We can test for that. We can see if that's true, and the first thing we have to do is disprove the null. We have to say, the chance of getting our data in a world where there's no correlation is incredibly, incredibly small. Because we've seen such a high correlation, for that to have appeared just by random chance is incredibly unlikely. And this is why having a large sample size is important, because chance events happen all the time. Say I have a D100, a 100-sided dice, and I roll it, and I say, gosh, this dice is random, it's very unlikely that I roll 100. But sometimes I will. Occasionally I'll roll 100. And with a sample size of 1, you can't really say, oh no, the dice is rigged, right? It's just, maybe I got lucky. But if you roll it 100 times and it always rolls 100, Now the chance of you actually rolling 100 is like 1 in a Google or something. Like it's maybe even lower than that. It's infinitesimally small. And so the dice is rigged. Like we can conclude, because the probability is like lower than the amount of seconds in the history of the universe, we can conclude that the dice is rigged. Same for us, right? We want to show that the data we're getting is so unlikely under sheer random chance that we can rule it out. And once we've done that, we can sort of float hypotheses for what might be the actual cause. And we think that the actual cause is because of the reasons we discussed, like wasps being a high trophic level, wasps being host specific, wasps being variably sensitive to ecological disturbance, and so on. Okay, so we get our statistical data. That's all good. What can we actually do if we have a result that suggests we could use them as a bioindicator? Well, we're going to need an ID tool. Even if we find that they are a great bioindicator, if we don't have a way to quickly get wasp samples from a study site, ID them, and then do a diagnosis, there's nothing like... Our idea is just kind of like a fantasy. So as I go, I'll be taking photos of the wasps and feeding them into a neural network that will hopefully produce a wasp ID tool. That's the goal. That's the final thing we want to produce. And that's basically what we'll be doing. This is a graph I made just using fake data. You can see all of these bars are identical. All I've done is scaled them so that there's absolutely no variation in like the ratio of blue to black to purple. It's obviously fake data, but this is kind of what we'd expect. So each color represents a family of wasps. We've got chalcidoids, we've got diapriids, we've got ichneumons. From site to site there'll be different numbers of them, and each site will have correlating vegetative data. A couple of years back we had massive flooding in summer, and I know for a fact that ruined some people's masters projects. So fingers crossed, no insane hurricane flooding, please. I would like some data to work with, but we don't get to pick what the weather has in store for us, do we? If we can get a plot that looks like this, I will be very, very happy. This is a scatter plot from a paper by Anderson et al. In 2011. And they were doing a very, very similar thing to us. They're kind of one of the inspirations for our study. So they were looking at pasture land, so not forest, pasture land in Ireland. But they were doing the same thing, right? They were measuring parasitic wasp abundance and diversity and comparing it to vegetative data, the abundance of other invertebrates. And this graph, the mirroring of my camera is causing me infinite concern. What we have is the x-axis here is the abundance of Parasitoid Hymenoptera. So if you were on 200, it would mean that at that site, when they brought their traps in, they found 200 Hymenopterans. Hymenopterans is the wasp, but it's also bees and ants. They're the other main groups of Hymenoptera. Technically, bees and ants are just derived wasps. They're just funny, unique kinds of wasp, but they get their own name because they are pretty unique. And then on the y-axis, we have the taxon richness of other arthropods. So this is non-hymenopterans. If you're a fly or a beetle, you go on the y-axis, and it's not abundance, it's taxon richness, which means the number of different taxa. So a taxa is like, so I don't know what taxonomic rank they're using. It could be species, it could be genera, it could be family. Whatever they're using, this is the number of different ones that they found. And what you can see is we have our regression line, which is the line going up, and that's showing that's the line of best fit, so it's minimizing the distance of all the points from the line, and it shows, in simple terms, as parasitoid Hymenoptera abundance increases, so does arthropod taxon richness. Now, that's really interesting, right? It doesn't tell us why. We have no idea if one's causing the other. Probably more likely is that both are being controlled by a whole set of environmental variables that are going on in the background. We also get a p-value. A p-value is, in simple terms, the chance that the data you found looks like this just through random chance, like I was talking about before. The p is less than 0.001, which means it's like less than a 1 in 10,000 chance. Probably much, much less, because we have a very nice correlation. Yeah, so if we can get something like this, I mean, this is from a published paper, I will be overjoyed. But I won't get too optimistic, because things can go wrong. Things can definitely go wrong. Here are some examples of things that might go wrong. The first is flooding. Flooding is not good. Actual flooding could wash the traps away, totally ruin the data. But even rainfall is pretty bad. If it washes out our traps, we really need dry spells. It doesn't have to be dry the whole time, because we're only putting the traps out for 5 days a month. But we do need blocks of 5 days with no rain. And we don't get to choose whether those exist or not. All we can do is cross our fingers and hope, and be ready to seize the chance when it presents itself. A second risk is the public, the general public. The traps, they're just loose, we just put them on the ground, and if we put them in a public space, like a regional park, then any number of people could come and take them, or mess with them, or turn them around. My supervisor has a story about a time when he came back and found that someone had done a massive poo in his traps and just left it there for him, which I think was quite wonderful to find. That's what you want. You know, you drive like an hour out of town to find your trap, and then it's just the data's ruined because someone's taken a dump in your trap. So I hope that doesn't happen, because that sounds pretty gross. But also people might steal them because they think that that's funny, or they might try to clean them up because they kind of look like someone's plastic rubbish. To combat this, last time I put some traps out, I did have one go missing. I suspect it was cleaned up by a member of the public trying to be helpful. So I made this sign here. It says, "Parasitoid trapping, do not disturb." And I tried to make it a bit scary. I was like, these wasps can't hurt you, they're fine. But they look kind of intimidating, and no one's going to really know what they are because they're so niche. So if I just say parasitoid trapping, people will be like, oh my god, there's parasitoids. There's parasitoids in the dirt. If I disturb them, I might get parasitoidized. Parasitoidized. They won't. But they don't need to know that. They just have to be afraid so they don't mess with my stuff. The third problem is a data problem. And the problem, I mean, this graph is not very helpful, but the problem is that what could happen is we do this research and we find a really nice correlation. We find that there are these wasp groups that are amazing predictors of diversity. So in this place with lots of diversity, you always find the wasp. Here, without the diversity, you never find the wasp. And we think, oh my god, like, this is amazing. We've got the perfect correlate. We don't know why. We'd have to do more research to find out why, but it doesn't matter. We can already start working on the bioindicator thing, because the bioindicator only needs correlation. The idea is just that we can diagnose. All this monitoring stuff requires secondary research to actually find out the cause of whatever trend we're seeing. But the correlation is enough to get started. So we have this amazing data set, and we take it to a different forest, and we try to apply the same principle, and then it doesn't do anything. Like, totally nonsense. And it turns out that what we'd found was not a genuine trend in the relationship between parasitoids and ecosystems. What we'd found was a specific information about the way the particular ecosystems we were working in functioned. And actually, there's no generalizable component. Now, there's some use still in being able to say this particular park can be diagnosed using wasps, but that's much less helpful than saying every single park can be diagnosed using wasps. We're not probably going to get a whole bunch of funding by saying we can tell you exactly what's there in this one park. I mean, maybe we'll get called back to do some monitoring of that park, but that's probably about it. This one is scary because until we've got the data and then done a future experiment testing it in a different place, we don't know which world we live in, right? I mean, it kind of inspires a thrill because you think maybe I'm wrong and all of this was for nothing, and we like to live on the edge. So here's the timeline of the wider project. I'm working in the early phase. I'm working up here. So the first thing to do is to demonstrate the bioindicator potential. We want to show that this is a possible thing. To do that, we do my experiment, we get this sort of predictive set, and then we want to test my findings in a new environment that we don't know anything about, and see if they hold up, see if they are accurate for diagnostics. Then we have our kind of prototype. What we need to do is refine the taxonomy, refine the ecological data, because having a prototype doesn't mean anything if we don't have a database of what each wasp does. And then once we've done all of that, we can make our integrated terrestrial bioindicator system. That's the end goal. Could take 20 years. I mean, it'll be not my work by then. I'm just a piece in the wider puzzle. But that's the beauty of science, right? You get to be part of something bigger than you. Yeah, so that's the project. Thanks to University of Auckland and Manaaki Whenua Landcare Research for supervising my study. Thank you to Auckland Council and the Department of Conservation for letting us work in their sites and use some of their information. And yeah, that's it. I'll turn over to questions to the ghosts, but I know that you guys don't speak in this way. If you do have any questions, leave them in the comments. I'm pretty bad at answering comments usually, but for this one, I will do my academic duty. If you are interested in this kind of video, that's cool. It's nice to see you. I'll probably make more, but these are mostly for me. I understand that will probably tank my channel in the analytics and ruin my chances at becoming a sort of Pokemon YouTuber. But I think for now, the wasps are my main priority. And if you did like the video and you are interested, I want to make more, and that's really great, because I think that science communication is really hard. It's really hard to get people seeing that science is just something you can do. You know, like okay, we have these institutions like the universities and the Crown Research Institutes and stuff that can seem quite constraining, but the actual things that we're doing as scientists are totally understandable, especially like biology, right? What I'm doing is putting out a piece of plastic in a field and then counting bugs. And like, you know, you could teach a kid to do that. I mean, you need to take them out of school and stuff and just focus on counting bugs for a while, which is probably not the way to come up with like a well-rounded adult. But you could do it, and it would work. And then you have to teach them how to use Python and a camera. But you could do that too, if you wanted. And then you'd create like Super Henry. They'd be like me, but like so powerful. And I'm already quite powerful. I'm on a full scholarship for this, so that's pretty good. Anything else to say? I don't think so. Thanks for watching. Have a good one. And yeah, see ya.